we will discuss seed, germination and its structure. Okay, seed structure and germination. What is the seed? Seed, it is a ripened ovule, and a seed contains an embryo and the endosperm. Embryo is the actual baby plant, whereas the endosperm is the food which is stored for the embryo. So that is basically what a seed is. Now, types of seeds, depending upon where this food is stored. Two types. Again, this is called albuminous and exalbuminous. Albuminous seeds have their food stored in the endosperm, and they're also known as the endospermic seeds. Example, the two types, monocot and dicot, barley, rice, monocot, and castor and cotton, dicot. Exalbuminous or non-endospermic seeds are those that store their food in the cotyledon instead of the endosperm. And the examples would be mustard, sunflower, pea, mango, etc. We'll just go on to the next one. Monocotyledonous seeds. They have a single cotyledon. They have a single cotyledon like this. Okay, single part. Whereas the dicotyledonous seeds or dicot seeds have two parts, two cotyledons. So that's all that there is. Okay. We'll move on to an example of a dicotyledonous seed. We'll take bean seed and its structure. The bean seed here is its structure. It has this kidney kind of shape, and this part has a prominent marking. This is actually the hilum. Okay, and it's called the scar left by its attachment to the placenta. The scar left where its attachment was in the placenta. Here's a tiny dot. It's a microscopic hole called the micropyle. This micropyle allows water to enter into the seed, and the seed is actually covered by a waterproof coat, okay, a cover known as the testa. It is waterproof and it also protects the seed. So let's just say it's protective in its function. The second layer, just beneath the testa, we find tegmen, which is a very thin, papery, let's just say it's a dry, paper-like cover. It has a lot of work to do in the process of germination, which we shall discuss later. So, a bean seed has the outermost cover made of testa. Just within, inside it, we have tegmen, which is a thin paper-like covering, which also has a protective function. And then on one side, it has a very tiny microscopic hole called the micropyle, and a big mark called the hilum, also known as the scar. And then inside of the what we call seed, we find the embryo. This embryo is the actual baby plant. This is the baby plant, and the plant has two sections. Okay, this leaf-like structure is known as the plumule. This grows into the shoot system, and this one is your radicle. CLE in bio. Okay, so this radicle is the one that grows into the root system. So this is basically the structure of your bean seed, and this cotyledon, this is the one that actually has the food which is stored for the embryo. Well, the reason why the food is stored for the embryo, this embryo in the process of germination needs to develop from an embryo into a seedling. So a tiny structure has to grow into a structure like this, leaves with roots. So that's a seedling. In order to grow, it needs food. So the plants make their own food in the leaves, but since it does not have leaves, how can it grow? So the food has to be stored somewhere. So it is stored here either in the endosperm or in the cotyledons. We move on to the monocotyledonous seed. Best example is your maize seed, and here is the structure. It is divided into two parts. This part is called the scutellum, which has the embryo. Okay, we'll discuss that later. And this part is the endosperm. Again, endosperm is the food stored for the embryo. And the endosperm has a very special layer of cells on its surface. I've drawn that layer. Okay, this is called the allelome. It's rich in protein. So we know that protein is required for growth, etc., etc. We'll deal with that again in the next chapter. Protein. Just understand, endosperm has a very special layer on the surface called allelome, containing large amounts of protein. And these two parts, the scutellum and the endosperm, is separated by a thin membrane known as the epithelium membrane. Now, inside the scutellum, here is the allelome. Okay. Inside the scutellum, we said it has, or it contains the, what we call, embryo. This is the plumule, which grows into the shoot system. This is the radicle, which grows into the root system. Now there are two extra structures in the maize seed. One is the coleoptile, which covers and protects the plumule, and coleorhiza, which covers and protects the radicle. So this is the difference between the seed of maize and a bean seed. Bean seed does not have coleorhiza, coleoptile. Okay? Now we move on to germination. The process of germination is just the development of an embryo into a seedling. Now, if you remember, embryo, as soon as it's formed, it does not begin to develop or germinate. It stays dormant. Normally, it stays dormant for a season. Okay? And then, in the next season, when it gets the requirements that are necessary for germination, only then it germinates. And the conditions that are required for germination is, first of all, water. It soaks the seed. And we talked about this earlier, where we brought up tegmen, which is a paper-like structure. Think of it as a blotting paper, okay, that dry paper. If it soaks in water, it begins to absorb water and swells up, and that breaks up this testa. So the work of tegmen during the process of germination is to absorb water because it's dry. So that's called imbibe, okay, or imbibition. The process of imbibition, it absorbs water, swells up, breaks up the outermost coat, outermost covering, or the testa. Now, microplant has no work to do because water can enter from any part of the seed. Okay, so that's the work of water, or that is why it is necessary, and it also soaks the food. The food that is stored for the embryo is a little hard, cannot be used by the embryo. But once water soaks it, it becomes soft, and the embryo can now use it. Air is required, especially oxygen is required in order to release energy. Energy is required for all the metabolic activities of the cell in order to turn itself from an embryo into a seedling. Then we need the right suitable temperature. Without suitable temperature, just providing water and oxygen does not make a seed to germinate. You can provide a seed with water, or you can just keep soaked seeds inside the fridge. You'll find that it will not germinate at all. So temperature, the right temperature. Seeds require a small range of favorable temperatures. Okay. So we'll move on to types of germination. There are basically two types: apigeal germination and hypogeal. Now, before I go on to discuss the types, let's bring up where this apigeal and hypogeal is. Okay. So this is the plumule, which grows into the shoot system, and this is the apex, or the part which is attached to the cotyledons. So between these 2 points, there's this structure known as the this is the epigeel. Sorry, not epigeel, epicotyl. And this is the hypocotyl. So these are the parts which grow rapidly in these two different types of germinations. That's the reason why one type of seed comes above the ground, the other remains below. Now we go to epigeel germination. Hypocotyl grows rapidly. I mean, this part grows rapidly. So if the seed is, let's bring it up here. This is the ground, this is the seed, and here is the radicle. If this part grows rapidly, it's going to form a bend like that. It keeps on growing, and the bend increases, this loop increases in size. So when it straightens out, it pulls the cotyledon out of the ground. So that is how the cotyledons come out of the ground. Because of rapid growth of hypocotyl, this part of the embryo. So this type of germination is called epigeel germination. And hypocotyl germination occurs in monocots mostly. And here is the plumule. This is the plumule which needs to go above. Okay. So in this case, it is the epigeel that grows rapidly. So if this part grows in size, it is actually pushing the plumule up. Okay, so it pushes the plumule right through the ground, and the seed remains on the ground. So the difference is this: epigeel germination, hypocotyl grows rapidly, and the cotyledons come above the ground. Whereas in the case of hypocotyl germination, it is epigeel that grows rapidly, pushes the plumule above the ground, and leaves the cotyledon or the endosperm or the seed on the ground. And lastly, we'll discuss vivipary. It is the germination within the fruit. Just inside the fruit, the seed begins to germinate while still attached to the parent plant. So if this happens, then this is it's called vivipary. Example would be mangrove seeds. Okay, so this is basically what the seed structure is and different types of germination.